says we're 20% of our final exam grade.
Yes, in other words, 5% of your overall course grade, or 10 if you replace a midterm.
So hypothetically, can we get more than 100 on the
No.
final? Like, not possible?
Absolutely not.
So this is 20% of our final exam grade and someone else is grading it?
Yes,
Okay.
according to a rubric.
Okay. That we will have.
I'll look at it. If it's really wacky, I'll step in, but it's like these elementary and high school rubrics, did student make point X? Yes, I don't want to do that. I may read your arguments for fun, but if you have any concerns about being fairly graded, certainly come to me on that. You'll know long before the final exam what the score, well, you'll know before the final exam what the score is. I will have instructions posted. I haven't finished composing the question yet or formulating the instructions. But this is in lieu, in preparation for rather than in lieu of. Next week's paper discussion in class is going to frame the issue. So the ethical question is essentially this. A couple of years back, virologists figured out how to turn an avian flu into an airborne human pathogen by fiddling with the hemagglutinin and neuraminidase genes, oddly enough. But it is essentially instructions on how to make a bioterror weapon. Because if they did it, they were into the science, the binding sites, yada, 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 wrote the paper, sent it off to the publisher, sent it off to the journal. The reviewers go, wait a minute, there's a concern here. So they went to the Navy, and the Navy said, absolutely not, don't publish that. Eventually it was published, so you'll get a little bit of the history of that. This is one of the papers, and it's a science paper, which means it's short and it's very short on details. But we'll bring this up as our discussion next week. I'll have the instructions for you. What you will write is only 2 pages. But I just want to frame the issue. Plus it's flu, so it's timely what we've been covering in class. But they used ferrets as their model system, so they deliberately infected ferrets with a crossover strain. Again, working out the biology, but there's an ethical question. There are a lot of ethical questions. It's a good thing to do. We learn more about the virus, but we could potentially create a World War Z scenario, yada, yada, yada. We'll be going down that vein. In that vein, this paper will be posted, but I'll show you how short it is. Lots of big pictures. But you guys like viruses, so
As much as you.
probably more. All right, I did want to finish. I gave very short shrift to the antiviral shtick yesterday, so let's briefly talk about that. So remembering that it's very difficult to treat viruses, the antivirals that are not vaccines for influenza on the market aren't very good. The 2 most modern ones, Tamiflu and Relenza, are neuraminidase inhibitors. So that works this way. Remember, as the new virion buds off of the host cell, it's still got the receptor-ligand interaction, which is very tight. So this is the hemagglutinin, sialic acid, or neuraminic acid, as your textbook calls it, bound. And it's bound very tightly. It's a high affinity interaction. Virus would not come off with any great frequency because of the affinity of this interaction. So the neuraminidase actually hydrolyzes the sialic acid, freeing the virus. So rather than dissociate, because the affinity is too high, it destroys the receptor and off it goes. And so these drugs, so this is sialic acid. It's a derivative of a carbohydrate. The structure of sialic acid on the cell surface is much more complex than this polymeric. But as you can see, at least with my eye, which is not that of a chemist, these don't look totally dissimilar to this. So the drugs, this is Relenza, I believe this is Tamiflu, were rationally designed molecules. What the chemists that were doing this were doing was looking at the structure of the neuraminidase and then trying to design a molecule that would fit into the active site. Unsurprising, well, not unsurprising perhaps, they did in fact succeed. And remembering back to your basic enzyme kinetics, oops, so basically I did it wrong. So this is the Michaelis-Menten expression of enzyme kinetics. Sadly, we're not going to spend a couple of days on this, but this is essentially a statement of an enzyme has to bind its substrate in order to catalyze the reaction and release the product. And so we've got an association and a dissociation, which is known as K1 and K minus 1. So those are two, we're governed by two rate constants. If we have a competitive inhibitor, that means the inhibitor will bind the enzyme and by doing so prevent the substrate from binding. This is as opposed to a non-competitive inhibitor that can bind the enzyme whether the substrate is bound or not. Different situation, both of them inhibit activity. And so, I won't go into the kinetics of this, but this is the general idea behind a competitive inhibitor. If your drug binds to the enzyme can't bind to its substrate, in this case sialic acid, doesn't cleave the sialic acid, can't bud off and go infect new cells. So we're inhibiting the enzyme by putting a small molecule in the active site, thus it's a competitive inhibitor. So here you have it. This is a space-filling model of neuraminidase, which so happens is a tetramer, and you see there it is. This is Rolenza bound to the active site. So we even have structural information about what the competitive inhibitor looks like. This is Tamiflu here. That's the generic name. Of course, this is a atomic detailed model. There's active site residues, the substrate bound, and you can generally see the general shape of the drug in the active site, and you could get a visual feel for how it would exclude the substrate. The new one, right, I ended up, Perimavir is not an approved, FDA-approved drug yet. It is also a competitive inhibitor. This is it here, but it's another class of it's a specialized type of inhibitor called a transition state analog. Which means it's modeled not to look like the substrate nor the product. Keep in mind, enzymes don't change equilibrium, so enzymes also bind products. You can design inhibitors off of the substrate or product, but rather like that transitional high energy state that is the substrate in the midst of its reaction to becoming product. So if it's a hydrolysis of a peptide bond, for example, it would be an intermediate state where the bond is partly broken. You can get some very weird geometries like 5 bonds on a carbon in a transition state that doesn't really exist for any length of time, so you can't really determine what it is. But the idea behind better antivirals here is more carefully approximating that transition state structure, and you get a better binder. And I just go back to a little basic chemical kinetics here. Entertainment, if no other reason. So for a spontaneous reaction, the free energy of the substrates has to be higher than the free energy of the products. Otherwise it won't be spontaneous. Basic thermodynamics tells us this, and we have a delta G, so a change in free energy between the substrate and the products. Or as with negative delta G equals spontaneous. Now, the free energy of a transition state, it's a strain geometry, it's very unstable, is very, very high. So in order to get from substrates to products, we have to go through the transition state. And enzymes catalyze their reactions in a number of ways, one of which is by lowering that free energy barrier so that substrates more readily go through the process. This is something called reaction progress, which means the free energy state as the substrates move through the reaction. It's not time, it's not concentration, it's degree of doneness, essentially. And so a catalyzed reaction will have a lower free energy of the transition, lower free energy in the transition state. It does that by binding to the transition state. So if you have a strained geometry in an unstable molecule, you can stabilize it by binding to it, providing some favorable interactions to lower the unfavorability of the transition state. So enzymes actually do that catalysis. So the gold standard for developing a drug that's going to competitively inhibit an enzyme is to make a stable molecule that looks like the transition state. And in doing so, more tightly bind to the enzyme. So if we had our binding curve, as I asked you to do on the, or actually that would be concentration of drug. We have a rectangular hyperbola approaching an asymptote Bmax. A better drug is going to look like that. A worse drug is going to look like that. So when you're a chemist and you're or you can be a biologist and do this too, when you're developing a new drug, you're generally looking for a lower KD, tighter binding. And we're achieving the tighter binding, the company that's developing the drug is achieving tighter binding by structural information about the enzyme active site, chemical knowledge of how to make the molecule that fits, and then binding assays that would show, gee, peramivir binds to neuraminidase, that's hard to say, much more tightly, and it would look like this. So when you're looking at data, you can look at the graph and say, well, it takes a lower concentration of drug to achieve any similar fraction of the population bound, that point versus that point. So in the transition state analog curve, we're going to get more binding at lower concentrations, which you can imagine as a biologist is good. Drugs have side effects. The less drug you have to use, the fewer negative outcomes there are likely to be. Plus tighter binding means higher affinity, which means less binding to other things in general. So we don't have a lot of good antivirals, but there are efforts out there, and there are basic scientific issues underlying the search for new drugs, transition state analogs. Actually, if it's an inhibitor, it's a transition state inhibitor. It's an analog of the substrate, or the transition state that inhibits the enzyme.
What's in parentheses about?
It's brackets, drug concentration, so it's in this case, it's what would be called the ligand concentration, what it is that's sticking to the molecule. So our assay for binding would be, we keep the concentration of neuraminidase constant, and we have different samples. Let me draw this bigger since. That's binding at equilibrium. So what you do is do several points. You maybe have a 96 well plate, and you have 12 concentrations, you do a triplicate, whatever the assay is. You mix enzyme and inhibitor, you wait for equilibrium to be achieved, and then you measure the bound. Lots of different ways to do that. But so you measure the amount bound at a given inhibitor concentration and a fixed enzyme concentration. We're not going to, I'm happy to do it in my office hours if you guys want to see the derivation of this stuff. Wow. You then fit to the rectangular hyperbola. That concentration of ligand at which binding is half maximal is KD. You measure KD in other ways, but in a binding assay, you do different points, wait for equilibrium, measure binding, fit the data. Your constant should explain, because it's the equilibrium dissociation constant. Given a known concentration, there are ways to take care of that you don't know which enzyme is free and which is bound. You get the equilibrium dissociation constant so that for any given enzyme concentration, if you know the KD, you know how much will be bound. That's quantitatively describing binding. And then something that has a lower KD, I'm not drawing the points there, a lower KD is literally a lower KD, right? The black KD is less than the green KD. It means the concentration of ligand it took to get to half maximal binding is less. Therefore, the affinity is higher. So chemists would get really, or drug companies would get really excited by this. Plus, I like it. Was that the answer to your question? You don't remember this?
Okay.
I was thinking about teaching when I got up this morning. I'm not wearing my formal teaching gear. All right, chapter 17, retrovirus.
As far as the quizzes go, are we following the syllabus?
Yes. So are we following the syllabus with respect to quizzes? Yes. As I've said before, I'm moving faster through this section of the course than planned, because I think the last section of the course is far more interesting. Quizzes are all due
When there is a
day on the syllabus. You can turn them in whenever you like, but up until the time they're due. The test will be on chapters 10 through 19, at least I think right now.
Yeah.
I'm not going to spring that on you, so we may finish a class or two early. The material and the dates will all be due, and the exam will occur on the date in the syllabus. I'm sorry if there was stress, and it seemed like there was something wrong with chapter 16 and 17 on the quizzes.
Yeah, they were grading it.
Send me what you got on 17. It'll be fine. The crappy textbook website quiz, I know. Beats me writing a quiz. Okay. So retroviruses. So there's a general chapter, and then chapter 18 focuses in on HIV. So covering the general, these are generally applicable. There'll be variations depending on the type of retrovirus. Have our viruses that undergo reverse transcription, so they make DNA from an RNA genome prior to packaging their RNA genome in new viral particles. So the discovery of retroviruses, which occurred really in the early 70s, that was the first crack at the central dogma of DNA always stores the genetic information and encodes RNA. Counterexample, the RNA can, retroviruses encode DNA and be the repository of the genomic information. They were originally isolated from either animals and then very quickly afterwards people who had leukemia. So a lot of the thought is that they are causative agents of leukemia, and retroviruses can indeed cause cancer. A lot of them were cultured from those cancer cells and may not have a direct influence on leukemia. So for example, the first human retrovirus isolated was human T-cell lymphoma virus. So, characteristics of a retrovirus: plus stranded RNA virus, 2 copies per genome, per virion. So it's not double stranded, but 2 single strands that are in fact base paired to each other. It's starting to get weird, isn't it? But are not complements overall. So you see in the representation from your textbook, there's a very short sequence of base pairing. So there are hydrogen bonds in that segment, but there are 2 distinct copies of the genome. They are spherical and unborn. Several.
Question.
Yes, please.
Are they both coding strands, both in the copy, or?
Yes. I had to do the plus decoding translation. That was the delay. Yes, they are not so if they were plus and minus, they would be complements and likely double. They would be the stable state would be double-stranded RNA. 3 classes of protein: matrix, capsid, and transmembrane or surface. So in the garden variety retrovirus, we have the viral envelope, the black line, the glycoproteins, which are surface. In the case of, the generalized is transmembrane with a surface protein tightly bound to that transmembrane protein. So it's 2 different molecules forming a complex. Not always the case, just the general case. In between the envelope and the capsid is a matrix, so that is proteinaceous. We then have the structural component, we have the capsid protein, and inside the genomes with associated nucleoproteins.
What was the S you did?
Surface. Figured out where the biology department keeps new dry erase markers. I apologize for the aging infirmity of mine. Let's see, this one looks better. Dry erase marker. The genomes are organized into 3 groups of genes. GAG, which are named for their antigenicity. Group-specific antigen. These are the internal structural proteins. Yes. These are the GAG genes encode the internal structural proteins of the virus. The Hall genes, as you may well imagine, encode enzymes. Most importantly, polymerase, which is reverse transcriptive. More about that. And then the E and V genes encode the envelope proteins, TMSU. So all retroviruses have the 3 classes of genes. Some of them have 3 genes. So we'll see with HIV the story gets more complicated. How's my speed? Too high? If only I didn't drink 12 cups of coffee. Great. Attachment and entry, same receptor mediated. We have the surface protein bind the host receptor. HIV, so this is GP120 and CD4. followed by fusion of the virus, and then emergence of a reverse transcription complex. Being retroviruses, of course, we have to go from RNA to DNA. Retroviruses are going to integrate into the host chromosome, so we go from single-stranded RNA to single-stranded DNA, to double-stranded DNA, to integration in the host chromosome, and then transcription and replication later on. So so there is a host from the prior host encoded tRNA associated with the viral genome in this reverse transcription complex. So we're dealing with a complex. There are both the enzymes reverse transcriptase and other associated proteins are present. Reverse transcriptase does what I think it would do. Transcribe RNA into DNA. Like all polymerase events, it's 5' to 3'. So if we think about this mechanistically, it needs, it goes 5' to 3', and it needs a primer. That primer is the tRNA. So what it needs, that tRNA provides the 3' hydroxyl, to which the polymerase, like other polymerases, catalyzes formation of a phosphodiester bond with the incoming nucleotide. So this is 5' to 3' of the genome. The tRNA is bound at the PBS, or primer binding site. So the first event in reverse transcription is polymerization of that short stretch of genome, the reverse transcription of the 5' sequence, or the sequence 5' to the primer binding site, which is only a minority of the genome itself. And a virally encoded enzyme present in the reverse transcription complex called RNase H hydrolyzes RNA from RNA DNA hybrids. So as that polymerization event occurs, purple is RNA. What color is that? Pink? Mauve? Taupe? I'm serious, what color is this? Brown? What do you want me to call it?
Taupe.
Taupe, okay. So we have a purple taupe hybrid. Is taupe really like brownish?
Kinda, yeah.
Okay, I'll take your word for it. I hope we're not fooling with someone who said taupe. All right. Squirrel. We have an RNA-DNA hybrid molecule. It's not a normal base pairing arrangement. We have the enzyme destroy the RNA in that complex, leaving us with a short stretch of DNA, and then the dotted lines there indicate that that's been hydrolyzed. The sequence that was reverse transcribed is duplicated at either end of the genome so that the new DNA can now prime the synthesis of the minus strand. I'm going to write that down. Destruction of the DNA-RNA hybrid, priming of synthesis of a reverse strand, and then of course, the minus strand, and then the minus strand is template for synthesis of the parental DNA. And then we have a displacement of I merged two events into one. Let me come back here. So we have synthesis of the plus strand of DNA. This is the RNA. We have the RNA priming, synthesis of this tail end of the plus strand. RNase H then can destroy the parental RNA strand, leaving us with a short stretch of DNA that's the 3' end of the plus strand that, because of the complementarity of the ends of the genome, can prime synthesis of the full plus strand. Ta-da. At either end of the genome, of course in the RNA, but in the DNA here we have an LTR, which stands for long terminal repeat. Because it's long, it's at the termini, they are at the termini, and they're repetitive sequences. That's recognized by another viral enzyme, integrase. The double, I also forgot to mention, the double-stranded DNA is known as the provirus. We have transport to the nucleus while reverse transcription is going on. Once in the nucleus, this circle here, by the way, this confused me for a long time. I'm like, that's not a vesicle. That's to indicate that's the reverse transcription complex. So a number of proteins and nucleic acids bound to one another. Integrase cuts the host chromosome, and DNA repair machinery of the cell thinks it's a nick in the chromosome and ligates it, so that you then have integration of the provirus into the host chromosome, which can either begin the active transcription and assembly of new viruses, or be latent for a period of time and divide with the cell as part of the chromosome.
Does the integrase just insert randomly, or does it put it in a specific spot?
So it's effectively randomly, and indeed that's many of there is some, I'm hesitating because there is some base pairing in that, but the way in which viruses cause cancer is by either turning on or off genes inappropriately, or sitting down in the middle of them and destroying them. That paper we talked about last week, where the ancient retroviral element served as an enhancer, is sort of the biological outcome of viral insertion that was favorable to our brain development, at least, as opposed to cancer. I'll have an example of that in a few minutes. So once in the chromosome, the LTR serves as both a transcription start and stop site, and the organization of the genes from GAG to POL to envelope is such that transcription from the DNA to RNA, the same molecule, can either be messenger RNA or the progeny genome. So it's a plus-stranded RNA virus. So the packaged genome is also the messenger RNA. So that's what this is trying to communicate to you, is that they're the same thing. Splicing of the messenger RNA occurs. So for the envelope proteins, the upstream elements are removed so this is for the upstream elements are removed as splicing, so we have transcription start site envelope proteins. That's what this is indicating here. Export to the rough endoplasmic reticulum, synthesis of the envelope proteins, which is, so that's TM and SU, glycosylation transport to the Golgi, as we've seen with other viruses. For the remaining GAG and POL genes, they're synthesized as a polycistronic fashion, so there can be multiple genes in here coding for multiple polypeptides. But translation occurs, there's a stop codon that is occasionally missed. It's about 95 to 5% where we get our group-specific antigens. We need a lot more of those. Those are the structural proteins of the virus, and many fewer of the polymerase, the enzymes. Right? Enzymes catalyze reactions are not present at this high concentration. So that by this run-through translation, the virus makes appropriate amounts of its protein. So, I'm sorry, let me say that formally. The envelope proteins are from a splice product. The GAG and POL genes are translated from a genome length RNA. So some of our messenger RNA are in fact the progeny genomes. The GAG POL proteins are one barystylated. So that's addition of a hydrophobic group that results in membrane anchoring. So as they're expressed and bind to one another, they also become associated with the membrane and the TMSU proteins. So these can be separate proteins, they can be domains of the same protein. So that's matrix, that's capsid, CA, and then nucleocapsid for the elements that bind to the RNA. So in this rendering of a general retrovirus, it's all the same molecule. The binding to the membrane and binding to one another results in a packaging of, so addition of more monomers of GAG and GAG-POL subunits result in formation of a capsid that of course buds off with the envelope containing glycoproteins as it exits the cell to go infect something else. There's the summation figure. Your book has a discussion about retroviruses as gene therapy. Imagine they might be a good target for gene therapy, right? If you can control what RNA is in the virus, that's relatively easy to do in the lab. You can synthesize RNA coding for what you want it to. It could then in fact insert into a chromosome and, for example, functional copy of a gene for somebody with a defective copy of the gene, cystic fibrosis, as I talked about at the beginning of the semester with the cold virus. The retrovirus offers that ability to deliver a stable copy of a gene. So it's not in a plasmid, it's not extra chromosomal. You have the virus itself as the tool to insert a functional gene. So there's an X-linked X-skid that your book talks about. I just wanted to flesh out some details here because when I was a kid I saw a people it was made for TV movie about this. So I've always remembered it. So the X-linked severe combined immunodeficiency results from disruption at a single, or results at a failure to form an immune system from a single single gene product, so it's a developmental issue. And throughout human history, that's more or less been incompatible with life. You're born, you get infected, you die. Starting in the 1960s, medicine started putting people with severe combined immunodeficiency into bubbles that were sterilized. So the most famous case of which is David Vedder, the boy in the bubble. Who lived 13 years. He was very mentally unstable, as you can imagine, because he never had any human contact. So he was born, immediately put into a sealed environment. There he is as a young kid. He was born in 1971. That's the other reason he would be my age if he were still alive. And his diapers were sterilized. Everything that went in had to be sterilized, and of course no one could touch him. And I don't know if that's either his father or his doctor there, and he's still inside a bubble. NASA built him a suit so that he could walk around outside his bubble. He used it 8 times, he didn't like it. He grew out of it. And eventually, so the way to treat these, the idea was they would keep the child in a bubble until such time as a bone marrow donor could be found. And so this kid never got a match. His sister, so it's X-linked, sorry, I didn't, what's in the name? It's X-linked, so women who are heterozygous for it are perfectly normal. His mother clearly had the marker, and he had a Y chromosome, so it happens in males. They were hoping his sister would be a bone marrow donor, but she wasn't a tissue type match. They could never find one, so the kid stayed in a bubble. You do a bone marrow, I guess it's a transplant, and they can kids can generate a perfectly healthy immune system from the donor cells. So finally, because he was there's some interesting case history with the psychiatrist and what it was going to be like for this kid to be a teenager living totally apart from human contact. And so his parents decided to do an unmatched transplant of his sister's bone marrow as they had planned to do. Things went wrong, he got sick, they pulled him out, and at age 13 he died of Burkitt's lymphoma. Which is actually something people with AIDS die from with some frequency. Of course, it happened very quickly because he didn't have a functional immune system, but later on it was found that his sister had an Epstein-Barr viral infection to which he had absolutely no resistance. So it's a good story. Well, it's a terrible story for him, but the nice thing is that people aren't putting in bubbles anymore, at least with any great frequency. Of it in years and years, not since that TV movie, 1985 or 6. I was thinking about the TV movie. Because I assume bone marrow registries have gotten so much better. There are many more people in bone marrow registries. Tissue matching has come a long way since the 1980s. But your book, the throw-off comment from your book is that these immunodeficiency has been treated with gene therapy on retroviruses, and indeed it has been, but the problem is the cancer develops in some of the patients, so that it's yet another point where viruses have let us down as gene therapeutics. So I pulled the paper from a decade or so ago that discusses, you can look at it if you want, discusses the different viruses and retroviruses used for gene therapy in severe combined immunodeficiency. And what you see here is relative to the cold virus, like that you tried with cystic fibrosis, when you do it with somebody with severe combined immunodeficiency, they maybe get a couple of weeks of presence of the gene. The advantage of the retrovirus is it sits down in the genome and becomes permanent in a way that other viruses don't. The problem is it turns on cancer. So here, Drew, is the real answer to your question when it sits down, so this is the retrovirus, when it sits down in the genome very frequently, no problem. There's a lot of DNA that's not coding regions, and it doesn't have any effect upon host gene expression. But sometimes, with some frequency, and remember, if we have an immunocompromised person whose immune system is not going to recognize a cancerous cell, that's a serious, serious problem in a way it isn't for people with immune systems. Your retrovirus sits down. These are those long terminal repeats. It's both the transcription start and stop site. So this upstream, and this is a host oncogene, so a gene that when overexpressed causes cancer. That LTR can serve as the promoter for inappropriate expression of the oncogene. It can happen in several ways. It can be an enhancer element, like that paper we read last week. It can be a transcription start site. This binds to RNA polymerase. And when it turns on the oncogene, either as a fusion to a viral gene or by itself, these are supposedly the introns, and exons, and these are the introns that are going to be spliced out. You get gene therapy, in fact, causing cancer. So that's the mechanism of why retroviruses at least yet are not yet very promising therapeutics because they kill you from something else.
Can they take those genes out of the virus?
Because the virus needs the LTRs in order to reverse transcribe into DNA and integrate into the chromosome. So, good question. So without the LTRs, we never get from RNA to DNA, and the LTRs are also critical for recognition by integrase for integration into the chromosome. So it's the properties of the virus via those LTRs that give it the ability to insert the DNA, so the promise and the peril of insertion. So, before we get into HIV, let me torture you with this week's paper. I chose this one, so this was one of the ones mentioned on TWIV, but it's biotechnology and focus. Right? It has a biotechnology focus, and I know there are some biotech leaders, so I thought I'd throw that out there. Of course, at least the essence, in my opinion, of biotechnology is developing tools with which to do better biology, whether that means drug development or understanding biological systems. In this case, we have, where are they? MIT, oh yeah, the Rock. If you want to go to grad school, this is smack in the middle of Manhattan. It's very, very well endowed, and they give you free housing. So you can go live in New York City, go to the Rockefeller if you want. So this paper developed a novel, sensitive, and fast method for detecting hepatitis C infection in living cells. When you read a paper, you start, of course, with the title, Real-Time Imaging of a Hep C Infection Using a Fluorescence Cell-Based Reporter Systems. And the other reason I chose this is we talked about fluorescence methods as virological methods, and so here you go, it's real. So we see that there's a troubled history of hepatitis C research. It's only recently that hepatitis C has been able to be propagated in culture. So the paper uses hepatitis C, HCV, CC to denote that you have a strain of virus that is propagated in culture. Monitoring infection involves bulk population assays and terminal processing of potentially precious samples. There's a problem the biotech's going to solve. Other methods have short downsides as well. So they've developed a technology, previously intractable applications, to visualize an infection in primary cultures. So that's another important point. And so using their system that is a fluorescence reporter with modern analytical methods, namely automated microscopy, can open new doors for research. So they're developing a tool for scientists to study Hep C virus in the lab. So first a bit about green fluorescence protein, or GFP. That's why I love biology. Things are named for what they do. Green fluorescent protein fluoresces green. It's from, thank you, I'm trying to think of the Latin name. What is it?
Victoria.
The genus is right. Well, there are a number of species, so you could well be right. It's a weird protein. It's small. It's tightly folded. It forms what's called a beta barrel, and has modified amino acids in the middle of that barrel structure. So here we have N to C terminus, that's the backbone. And in the structure we have a modified chemistry, so this is just the tripeptide sequence. But in the protein, we've had some covalent chemistry happen, so we have a modified amino acid, and it's this group that has the property of fluorescing. So it's it's excited at, I forget what the wavelength is, it depends on the fluorescent proteins we use. Excited at one wavelength of light, fluoresces green. Very valuable to a microscopist because of its nature. It's small, it's fluorescent. You can attach it to other proteins and generally not affect their function. Something you have to worry about, but you can stick it on the inner C terminus of a protein and make it fluorescent, and it being fluorescent means you can watch it. People are doing all sorts of wonderful things, like creating fluorescent animals. You can image in vivo. This is part of a rat brain that's alive. A rat or mouse, I forget. You can label specific cell types in complex tissue. So this is a neuron and other cells around it. You'll note that those are different colors. Oh, sorry. More transgenic pigs. Glow in the dark. I love the yellow nose.
So it really can't have green eggs in ham?
You could have green eggs in ham, it'd just be fluorescent green. So he's done a bunny, yada, yada, yada. It's, there's no end to it. This generally happens in Asia, where they have much freer reign with animal research than we do. So the pigs are Korean. There are point mutations that have been made in the fluorescent protein, and there are other proteins, in fact, that other than the specific green fluorescent protein, that have given researchers a spectrum of fluorescent colors. So if you look at GFP, that's what it looks like, but there's cyan, blue, there's tangerine even. They're all different colors, they all fluoresce at different wavelengths, they're excited at different wavelengths. But the cool thing is you can have multiple fluorescent proteins in a sample and track them all separately. So we're going to see some of this in the paper here, namely, I think it's red fluorescent protein, RFP, and GFP. So, hepatitis C encodes, hepatitis C encodes a protease, not unlike the interferon, the influenza. Where did I put my green markers? No. Hepatitis C encodes a protease. Doesn't really matter for our purposes. It's NS3-4A that has a host substrate interferon beta protein stimulator 1, or IPS1. This is involved in host response to infection, so it very much parallels that influenza protein that cleaves the host protein that binds, that starts the signal that results in the host protein destroying the viral protein. So Hep C encodes a protease that destroys a protein that's involved in the response to Hep C. In other words, it's a proactive defense against the host. So they're going to use this protein, IPS1, as some common structural features. It's the substrate for that protease. That protease cleaves at residue 508. That's what that little triangle means. So they're going to take part of the protein, do some molecular biology, put GFP on the N-terminal end of the domain of the protein that gets cleaved. That domain has a transmembrane span, or transmembrane domain, so it's normally, this is mitochondrial, in the outer mitochondrial envelope. When cleavage occurs, of course, that transmembrane stays in the membrane, the rest of the protein is ready to go somewhere else. These researchers are attaching a fluorescent protein anchored in the membrane, and they have their cleavage site, and their assay is going to be, does the green move? They also have a variant that's red. It has a nuclear localization signal, or NLS. So when cleavage occurs, this is going to go to the nucleus. So in figure 1b, this is just our system works. This is a liver cell line, so it's hepatitis C, so this is the model cell line. Nuclear stained blue. And then with either, it's false colored, the green fluorescent or the red fluorescent. So there's no fluorescence here other than the nuclear stain. So there's nothing there, but we see a fluorescence outside the nucleus. The nucleus is blue. We have fluorescence there is emerged, so that's green and that's red, even though it's yellow. So this is their demonstration that their fusion proteins localize to the mitochondria. So this panel is infection with virus, either in cells with the wild type IPS or with a point mutation that results in a change in that residue 508 from cysteine to tyrosine. It's uncleavable. And so we have either the cell line or cell lines infected with altered viruses. These are just the names of them. And what you see is when we have wild type, that punctate fluorescence resulting from mitochondrial localization becomes diffuse. In other words, this gets cleaved, the green fluorescent protein is free to freely diffuse about the cell, unless, of course, it's the variant that's uncleavable, in which case it doesn't. And then these are two different strains. They're different strains of hepatitis C, so that their assay is named HDFR, or hepatitis C dependent fluorescence relocalization. So their sensitive assay is living cells, drop a virus in, some period of time later, they do time courses next, you get a change in the fluorescence pattern, so that this becomes the marker for that cell is infected. So in panel D of that figure, we see some elaboration on that. So we have the red fluorescent variant that in uninfected cells, so remember when it gets cleaved, this one, the red fluorescent variant has a nuclear localization signal. So when it's not infected, the mitochondrial localization is triumphant. When it cleaves, that nuclear localization signal is effectively activated, and you get the red fluorescent protein going to the nucleus. The story is more complicated than this, and in the interest of time, I'm going to move along. Well, what we see is red fluorescent protein in uninfected, that's mitochondrial localization. By the way, mitochondria are all over the place, that's why you're seeing this pattern. And then cleavage and just buffer, this is phosphate buffered saline, it's what these other things are going to be put into the cells in. Upon infection, we get localization to the nucleus. So that's the assay for infection. And then interferon beta, if you then put transfect the cells within to have a much higher concentration of interferon beta in the cell is then able to essentially fight off the infection, which is measured by the fact that there is no nuclear localization in the presence of high amounts of interferon beta. Same is true with an antibody that binds the protein so that you get, this is an antibody outside the cell. We see that we do get localization. That alone. So we get nuclear localization upon cleavage. They measure it in a different way. This is the nuclear stain down here as a control. So you can see that, for example, that's the nucleus, that's red fluorescent protein locating to that nucleus. So these are imaged at the same time. Another advantage of the modern microscopy is you can look at different wavelengths of light through your sample simultaneously. So now what in figure 2, I think in figure 2, what they're doing are time course experiments. So in panel B, we're looking at 0 to 24, a few more hours, but starting from the time point of infection. And then these images are started after a day. So this sample would be 26 hours in question.
What exactly inhibits the localization to the nucleus?
The mitochondrial transmembrane segment is predominant. So, thank you, good question. Transmembrane segments are very hydrophobic parts of proteins, so the free energy of getting them out of a membrane is very, very high, meaning very, very unfavorable. So they'll stay in a membrane. Nuclear transport requires movement through a hydrophilic environment. So as long as that membrane segment is attached, it's just observably true, the protein stays in the mitochondrial outer membrane. But it's upon cleavage that right here, so we're cutting away the transmembrane segment from the fluorescent protein and the nuclear localization signal, then that nuclear localization signal can actually localize the RFP to the nucleus. So time course post-infections, they're elaborating on the utility of their assay to study HCV replication cycle, in that from 0, you see signs of infection very early on, so that's touted as an advantage over other methods that take a lot longer to detect an infection, working with very small amounts of viral protein at first. But then as time goes along, we see more and more localization to the nucleus such that you have very clear infection later on. CMA is an inhibitor of the RNA-dependent RNA polymerase, so it's going to inhibit replication of the viral genome, and indeed you see no sign of infection in cells that are grown in the presence of that inhibitor. In a later time course, so we're looking at infected cells a day or so after infection, grown either just in dimethyl sulfoxide, which is the solvent for VX950. VX950 is a protease inhibitor that inhibits NS3-4A, so it inhibits the viral protease. And so that's added after infection is well established. It essentially, you can see, destroys the infection. We have the nuclear localization going back toward that punctate pattern. Further evidence that their reporter system works. The host cell receptor is CD. The host cell receptor is CD81, so the CD is constant domain. We'll see with HIV, the HIV equivalent would be CD4. So this is the host protein that is the receptor for the viral attachment. So they use a technique that's an inhibitory RNA technique called, I don't know, SHRNA. Short hairpin RNA. So it's a knockdown technique. By transfecting the cells with synthetic short RNAs, they disrupt transcription of the CD81 gene. So we're making cells that are identical to the strain and the cell line in every way except they express much less CD81. So they have both the green and red markers here in different, so cells transfected, two different cell populations, one transfected with the red with the nuclear localization, one with the green. So they were worried about detecting HCV in tissue that's complex and has cells of different origins in it. So the liver would have adipose cells and hepatocytes in there. So we have both red and green. The green are going to be CD81 knockdown. So IRR is an irrelevant RNA sequence. That's just a control that putting the RNA in there alone doesn't have it. And what we see is CD81, so the green spreads out when the virus infects. With CD81 knockdown, we see much lower degrees of infection, and it doesn't do anything to the red cells. So when you bring the reds back in, they have CD81. You see a differential degree of infectivity in that the red cells are still getting infected and localizing to the nucleus. In the complex population, the CD81s are not. So looking at this, they say, well, we can look at a tissue and look at a specific type of cell within a tissue and do the assay for viral infection.
So what does it do exactly? It does what to the CD81?
So the RNA, this is an inhibitory RNA technique, or you may have heard of as RNAi. The simplest form is you transfect cells with the complement to the messenger RNA. so there's a lot of it going in. And so you have messenger RNA that would normally be translated into protein, but if the complement is present in the cell, it's not transcribed in the cell. You get a double-stranded RNA, translation doesn't occur, the protein level goes down. So this is a lot easier to do than a knockout. So knockout is where you do molecular biology and go through the germline and raise multiple generations and have a mouse or another organism that's a knockout. So my favorite being the mouse without cannabinoid receptors, so they can't get stoned. I'll tell you that story someday. But that takes a long time and a lot of money. Whereas a small hairpin, an RNAi experiment can be done in, if you design it and do it well, it can be done in a couple of days for very little money. So it's not as ideal as a knockout. CD81 may well be necessary for the cells to grow in culture. It's a protein-protein, or cell interaction. Protein. Sorry, I stopped speaking in the middle. CD81 is important in cell-cell context. So you may not be able to knock it out and have the cell survive. I'm going to skip over that. Suffice it to say we have a marker for stress granules, and that cells get stressed when they're infected. I'll abbreviate this since I long ago lost most of you. And then they take primary hepatocytes, so these are liver cells that are not immortalized cell lines. Everything they've done thus far has been in an immortal cell line, so it's a cancer cell. It's not normal. You worry about developing things in immortal cells that don't work in primary culture, and they're interested in primary culture from, say, a medical diagnostic point of view. You want to be able to take a biopsy from a patient and see if there's HCV in there. And indeed, you see, once again, you have the punctate and diffuse following infection and then quantitation there. Shoot, I'm out of time. Wow, time flies when you talk about papers, doesn't it?